So in the last video I had discussed on the development of Oedogonium as well as the Antheridium, right? So in this particular video I am going to discuss on the fertilization. Now the process of fertilization, it is similar in both the Macarandrous as well as the Nanandrous species. Okay, and here what happened, if you remember that the antherozoids, they will come in contact with this gelatinous substance and they will enter here in the receptive spot of the Oedogonium. Okay, so fertilization will take place between the antherozoid and the egg. But here in this case, the antherozoids, how they reach the Oedogonium here, how the antherozoids, they reach the Oedogonium, they are attracted chemotactically. Okay, due to the chemical stimulus, these antherozoids, they will be attracted in response to the chemical stimulus. Okay, they will be attracted to the Oedogonium. So they are attracted chemotactically towards the major Oedogonium, and if you remember, there are usually two antherozoids are formed, right? Two antherozoids are formed in a single antheridium, but usually only a single antherozoid will enter in the Oedogonium. Okay, enters in the Oedogonium through this opening here, through this opening, that is the receptive spot which is present on the Oedogonial wall, on the wall of the Oedogonium. And what happen if more than two antherozoids they enter? Okay, usually only one enters through the receptive spot, and if more than one antherozoids enter the Oedogonium, only one antherozoid will fuse with the egg nucleus to form the diploid zygote. So when these male and female gametes, they fuse together, they will give rise to the formation of a zygote. So now we will take a look at the zygote formation. Now soon after fertilization, the zygote, it starts to separate from this Oedogonial wall. Okay, this is the Oedogonium and this is the zygote. These, they fuse together to form a zygote, and this zygote, it will start to separate from the Oedogonial wall, and it secretes a three-layered wall on itself. Okay, on its own. Three-layered, you can see here 1, 2, 3 in figure C. So it secretes a three-layered wall of its own, and a newly formed zygote here in figure C, you can see a newly formed zygote. It contains chloroplasts and hence it is green in color. But as it matures, there is accumulation of a reddish oil in the protoplasm, and it becomes, due to that reddish oil, it becomes reddish brown in color. Okay, so this is the process of fertilization. Now coming to the germination of zygote, how it germinates. The zygote is liberated, they are liberated by the decay of the Oedogonial wall. So the Oedogonial wall, it will become decayed and release the zygote. Okay, and this zygote, it will then settle down at the bottom of the pond, and at that time when it settled down at the bottom of the pond or lake, it will undergo a period of rest. Okay, a period of rest, that is the dormant period. We call it a dormant period. Okay, it will undergo the period of rest. Now this dormant state, it will help the zygote in tiding over the unfavorable conditions. Okay, so if the conditions are not favorable for the growth, for the germination of the zygote, then it will undergo a period of rest, that is the dormant period. Now suppose the favorable conditions comes, then the zygote will start to germinate. So the zygote, it germinates on the favorable conditions, and the diploid nucleus of the zygote, you can see actually this diploid nucleus of the zygote, it divides by meiosis to form 4 haploid nuclei. Now you can see here 1, 2, 3, 4. Okay, from a single nucleus it divides by meiosis, okay, to form 4 haploid nuclei, and followed by the division of the protoplast also, okay, into 4 segments. So you can see in figure, see here the protoplast also, it not only the nucleus, okay, not only the nuclei, there are 4 haploid nuclei, and also the protoplast also starts to divide into 4 segments, and each uninucleate segment, it will then develop, here in figure D you can see, it will then develop into a multiflagellate zoospores. You can see these are the flagella, okay, multiflagellate zoospores, and these zoospores, they are then liberated after the rupture of the zygote wall. You can see the here the wall is 3-layered, but here it already ruptures, releasing these zoomeiospores into the outside environment. These are now referred to as the zoomeiospores, which are formed after the meiosis. Okay, these are haploid, and these spores now, they will behave as asexual spore. If you remember, in asexual reproduction we had studied the formation of the zoospores. So these also, they will behave as the spores, the asexual zoospores, and these zoospores, they swim for some time, and after swimming for some time they will get attached to the substratum, okay, by their anterior end. Just remember that they will attach to the substratum by their anterior end. And germination of zoospore results in the formation of a haploid filament. You can see here, because each spore here is haploid, okay, formed from the diploid nucleus, undergo meiosis, we get 4 haploid spores, and these haploid spores thus will give rise to a haploid filament. Okay, so this filament is haploid. And in macrandrous dioecious, if you remember, dioecious meaning the male and the female, they are born on different filaments. So in that case, 2 of the spores, they will 2 of these zoospores, they will develop into male filament, and 2 of these, they will develop into female filaments. But in case of monoecious, where the both the male and the female, they are born on the same filament, so they will just give rise to a young haploid filament, and later on we know that the male and the female, they will develop on the same filament. But here I am talking about the dioecious species, okay, where in dioecious, you know that the male and female, they are born on different filaments. So that is why, just remember, 2 of these, they will give rise to a male filament, and 2 of the zoospores, they will give rise to the female filament. Okay.